Circuit Buddy. Today we'll be discussing about DNA replication. DNA replication is a very important topic of class 12 biology. It's also important for Neet. So let us discuss about DNA replication in details. In this video lecture, we'll be talking about the various concepts of DNA replication, the various enzymes which are involved in the process, and the experiments which were conducted in the past. Mainly the Meselson and Stahl experiment 1958. Okay. Now, what does it mean by replication? Replication comes from the word replica, which means exact copy of something. So, what does it mean by replication in short? It is the duplication of DNA, that is the synthesis of new DNA molecules from old DNA molecules. Okay. Now, let's talk about the central dogma of molecular biology. The central dogma of molecular biology tells us that from DNA we get RNA, and from RNA we get protein. The synthesis of RNA from DNA is called transcription, whereas the synthesis of protein from RNA, mainly the mRNA, is called translation. Okay? You can see here that new DNA molecules can be synthesized from old DNA molecules, and this is the process of replication. In DNA, we have 4 nitrogenous bases, and that's very important: adenine, guanine, thymine, and cytosine. Adenine and guanine are purine bases, that is, they have a heterocyclic ring structure, and thymine and cytosine are pyrimidine bases. They don't contain the heterocyclic ring structure, they are much simpler versions. Okay, now adenine is bound to thymine by 2 hydrogen bonds, and guanine is bound to cytosine by 3 hydrogen bonds. And one specific way to remember the number of bonds in between the specific base pairs is that C is the 3rd letter of the English alphabet. So you can easily remember from these that in between guanine and cytosine there are 3 hydrogen bonds. Okay. Now we are going to talk about the significance of nitrogen atoms within these nitrogenous bases, and that is the main reason behind this. Okay, so if you just talk about the significance of nitrogen, let's talk about the structure of adenine, which is a purine base. You see that there are several nitrogen atoms here in specific positions. Okay, in position 1, 3, 7, 9. Okay, and that tells us clearly that nitrogen is very important for the formation of nitrogenous bases like adenine, guanine, thymine, and cytosine. So now we are prepared to understand the Meselson and Stahl experiment. What they did was that they grew E. coli cells in a medium of NH4Cl or ammonium chloride, where the nitrogen was N15 isotope, or it is also called the heavy nitrogen. Okay, why is it called heavy? Because its mass number is slightly more than the normal N14. Okay, so actually mass number means the total number of proton and neutron. In case of the N14 we have only 7 protons and 7 neutrons, whereas in case of N15 we have 7 protons and 8 neutrons, which makes it a little bit heavier. Okay, so you see that they took E. coli cells and they grew E. coli cells in ammonium chloride solution containing N15 nitrogen for many generations. So after many generations they separated heavy DNA. Okay, they separated the heavy DNA and they transferred the heavy DNA into a medium of ammonium chloride solution containing the N14 type of isotope of nitrogen, or the light nitrogen. Okay, so what did they see here? Suppose this is the heavy DNA molecule. Okay, now in the first generation they saw that all the DNA molecules that were synthesized were an intermediate type of DNA, and they called it hybrid DNA, okay, because it contained both the N15 containing strand and the N14 containing strand. So it produced intermediate band in the centrifugation tube. Okay, now after that, in generation 2, they got a very significant result. Okay, so what they got is that you can see that this strand comes here, this strand comes here, but here the new strands were all N14 containing strands. Okay, so this is an N14 containing strand, this is also N14 containing strand, okay, or DNA. But here you can see that this is the hybrid form of DNA. So you can basically say that 50% of the DNA are purely light DNA. Okay, so they are light DNA molecules, and 50% of them are the hybrid DNA molecules by just fusing these mixture in the centrifugation tube. And in the centrifugation tube we had cesium chloride. Okay, because it has a concentration of 1.71 gram per cm3, which is nearly equal to the concentration of normal DNA containing only the N14 type of nitrogen atoms. Okay, so it clearly says us that DNA replication is not a conservative process, it's not a dispersive process, it's actually a semi-conservative process. Because if it is not true, then why is there these hybrid bands or hybrid DNA molecules? Okay, now this is the Meselson and Stahl experiment, which clearly proved that DNA replication is a semi-conservative replication, and after that Taylor et al. Also proved that it is a semi-conservative replication. And that's all about this first lecture. In the second lecture we'll be talking about the polymerases, their structures, their respective genes. Okay, and thank you very much for being with me till the end of the video. Have a great day.